This year's Anugerah Tokoh Penyelidik for the 24th Medical and Health Research Week goes to Congratulations to His Excellency Professor Dr. Raja Effendi Raja Ali. Professor Dr. Raja Effendi Raja Ali is a professor of medicine and previously held the highest position in the faculty as the Dean of Medicine. His research interests include inflammatory bowel disease, colorectal cancer, and the gut microbiome. He's been awarded with numerous research grants from Malaysia's Ministry of Higher Education as well as the industries. He's had a string of graduate students and has made significant contributions to the community through his involvement with various societies. Now, with that, ladies and gentlemen, the floor invites the Deputy Vice Chancellor, accompanied by the Dean of Medicine, as well as the Chairman of the 24th Medical and Health Research Week, to present the coveted Anugerah Tokoh Penyelidik for the 24th Medical and Health Research Week. We humbly welcome as well the Professor Raja Effendi Raja Ali, to the floor to receive the special award. Congratulations, His Excellency, on receiving the prestigious award. We invite the Professor to be ushered back to his seat. Right, so moving forward, it gives me great pleasure to welcome the Honorable Professor Dr. Tan Giok Chin, the Deputy Dean for Research and Innovation, to the rostrum to officially introduce our Tokoh Penyelidik, Professor Dr. Raja Effendi Raja Ali. Please extend a hearty greeting to the Professor. Thank you.
Thank you, Dr. Lokman. So a very good morning to everyone, Dato', Professor, doctors, and participants. First of all, I would like to thank the organizing committee for giving me the opportunity and the honor to introduce the recipients of the Tokoh Penyelidik Awards for the 24th Medical and Health Research Week 2022. As you have seen in the montage just now, the previous winner has all been from the preclinical. So this year is special. This year's the winner is a clinician, and I hope it will be an inspiration to all clinicians to work and become the next recipients of the Tokoh Penyelidik Award for the subsequent years. It is a pleasure to introduce to you Professor Raja Effendi Raja Ali. He is a clinician scientist, a gastroenterologist by profession. If you look at his citations, he is one of the fastest rising citations in as a clinician in our faculty. His citations at the moment in Google Scholar is more than 1,000. Professor Effendi graduated with honors degree in medicine and completed his higher diploma in clinical teaching, Master of Clinical Science, and Doctorate of Medicine from the National University of Ireland, NUI. He completed the higher training in gastroenterology and hepatology and was awarded the Certificate of Completion of Specialist Training by the Royal College of Physicians of Ireland. He is a Fellow of the Royal College of Physicians of Edinburgh and American Gastroenterological Associations. He's an active researcher with multiple grants being awarded that include the FRGS, the Fundamental, the Transdisciplinary National Grants, the RGS, numerous industrial grants, and international research grants as well. His research is mainly on inflammatory bowel disease, colorectal cancer, irritable bowel syndromes, and gut microbiome. He has accumulated 39 masters, 9 PhD students, and 10 clinical fellows in gastroenterology and endoscopy. He served as the immediate past president to the Malaysian Society of Gastroenterology and Hepatology. The list actually goes on and on, but I'm going to mention the last few. He has also obtained the professional certificate of the Oxford Executive Leadership Program and an MBA at the International University Malaya and Wales Partnership. So if the list of achievements, if I'm going to go on and read about it, it will take another maybe 10 minutes. So without further ado, I welcome Prof. Effendi, the recipients of the Anugerah Tokoh Penyelidik, to deliver his inspirational talk. The floor is all yours, Prof. Effendi.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Yang kita hormati sebentar tadi, Prof. Dr. Datuk Wang Kamal Mujani, Timbalan Naik Chancellor Research and Inovasi, Professor Dr. Halim, Company Datuk Timbalan Naik Chancellor downstairs, Dekan. Thank you for the lovely introduction, Prof. Tan Giok Chin, the Deputy Dean for Research and Innovation. I can see a lot of familiar faces physically here. Prof. Dr. Marina, Deputy Deans, Prof. Nur Azlina, Assistant Dean, Prof. Muhammad Maya, and also Prof. Nur Laila, maybe disappear now. Oh, at the back, sorry. And participants, and of course, very important, the chairperson, Prof. Dr. Wan Aslina Abdul Halim, the chairperson for the 24th Research Week for our faculty, and the organizing committee, and of course, sahabat baik saya, Prof. Azmi. I don't even know, I think he just retired and enjoying paid leave now for 3 months. And then, of course, the technical person are very important in this agenda. First and foremost, I would like to express my utmost gratitude to the panel selection to choose me to be awardees of this year, but it's humble and honor, and I think a lot more other people deserve this award. And I try my best to do some reflections and what lesson that I've learned for the last many years, even before joining UKM, and something that close to my heart, the diseases that I not only treat the patient, but also actively engage in research for the last number of years. This is my disclosures. Perhaps the very last one, some of the slides taken from my students and co-researchers, industry, and then my collections for the last more than 2 decades or so to come out with the presentation today. And I hope I can entertain you and try to finish as fast as possible. So what's going on today? Actually, happy 10 Muharram Day for the Islamic calendar, New Year, and a lot of barakah, if you know that. And also, over the weekend, the Terengganu football players beat convincingly penalty the Selangor Football Club, and we're going to see JDT in the final FA Cup for those who love football. Now, what are the menus today? A little bit introduction and the history, the journey of as a researcher, and of course, the main course is inflammatory bowel disease, plus other side dishes, probiotic, gut microbiota, IBS, fatty liver, and also to mention something citing current research that I've been doing, and a little bit about the research leadership. Now, I spent more than nearly half of my years since the age of 19 in the Republic of Ireland, and a lot of colleagues as well working here. And if you look at the similarity of the anatomy, architecture of the Peninsula, Malaysia, even the Borneo is more or less the same. This is like a memory. So I was located in the University Hospital here, and weekly always visited the clinical research facilities to join the scientists, molecular biologists, anatomists, a lot of interdisciplinary team to discuss many areas of diseases embedded together, not only in clinical aspect, but many other aspects, including psychological disorders in these many years during the training time. So my patient is in gastrointestinal disorders. You can see here the anatomically from the mouth to anus. Basically, we learn for the medical school, the upper GI tract, the lower GI tract, but also don't forget the middle GI tract, using all my toys here to Google and get all these necessary tissues, and of course, using the capsule also to see all these beautiful 7-8 meters long of the small bowels, and then can do various studies through the artificial intelligence nowadays and many other aspects of the research. Now, these are the reading books in early days, and by far the favorite one is by Katie Barker. Those who are doing the laboratory navigation, I think it's very useful to teach or to learn about various, not only basic, but also advanced molecular biological techniques, and various other books, and very important that written by actually urologists, how to succeed as hospital doctors from the very young age group, and how to convert the research into the manuscript and teaching the students during the round. Now, if I would like to summarize my life cycles, or perhaps your many other here physically or virtually, the life cycle when you start, this is what we call during the Jahiliyyah time, where I play a lot of footballs, I suppose, for 10 years or so, and you miss your prayers, and you work for football, you want to get money to support yourself, and to cut the story short, doing A-level and then studies. And perhaps the earliest mentors that I have, actually, weird enough, first year, look at the notice in the clinical science door to get the research assistant with the consultant clinical biochemist, Dr. Helen Grime, and worked in the lab in the summer for many summers, and introduced many technique which I have no idea at that time, ELISA, multiplex, and HPLC, and using the SMAC machine, a simultaneous multi-analyzer channel machine, to come out with a full blood count and renal profile, whatever that you do in the lab here. And as time goes by, that doing the internship and all these usual thing, a lot of obstacles need to be managed, and many mentors, and they are by far have many neurologist mentors, endocrinologists, and many such specialties, and encourage me to do what need to be done, and then also proceed. And by time, not many long ago, I would say not even 10 years now, I joined UKM on the 9th of March 2013, and start the career as a clinician research and also other things. So at that time, during the interview, I have to thank Professor S. Fadila, head of the Department of Medicine at that time. I didn't present this. These are the one that I put what I bring to the post to the UKM when I started joining 9 years ago. What are the value proposition canvas in terms of things that I have at that time in terms of to build up the or to get the running into the Department of Medicine and the Faculty of Medicine. In terms of teaching experience, the one that I love so much is about OSLE, or you can call here, we are familiar with OSCE, but also OSLE, Objective Structures Laboratory Examination, and of course OWP, Orchestrated Rhythm Performance. How do we do that over the time, and various teaching strategies and academic experience that I can bring to the universities when I joined UKM. And you can see here a very embarrassing research output, not that great, a very low H-index and citations. I need to work harder, and at that time no single invitation to review any article related to the disease that I work. So for virtual participants, those who postgrad studies, I can imagine these are the flowchart or the life cycle that you're going through after you graduate and you're doing some postgraduate studies, and you're going to do further on and on, contribute back to the society in the future. Now, during my course of the training, if you can see here, under the Royal College, there are a minimum of 2 to 3 years that you can do either MD or PhD in the lab-based or epidemiology or whatever that you like, depending on your sponsorship. And I engaged and decided to do that. Initially, it wasn't great, but as time goes by, I enjoy it so much because you can see a different perspective of diseases, of many other aspects of life when you stay in the lab. And in the earliest days before joining faculty here, I produced not many papers, mostly predominantly in the arena of inflammatory bowel disease, with a lot of peoples in Ireland and abroad, also come out with some other related book chapters, so and so, prior to joining here, and come up with the 2 theses. And the first one, nothing related to the IBD at all. It'll go low gamma GT in the Gilbert disease, subsequently venturing into the inflammatory bowel diseases. Now, I also bring an award to the UKM here, these postgraduate award winners in the presentation, and also the best non-consultant clinical teacher award many years ago. So the last day of endoscopy, and still taking colonic biopsy for my student on the last of February 22nd, 2013, before coming back to Malaysia and leaving behind many mentees, and they are much more successful than me. I'm very proud of all of them nowadays, and behind and contacting via the emails, and continue to get the feedback from them. It's very important. And I emailed Dr. Yui yesterday on Friday, and he got a massive writing to me, and thank you for all this the previous mentorship that given to him for the last couple of years, and now becoming consultant in Ireland. He's from Sungai Petani, and Clifford from Kota Kinabalu. Now, prior to joining here, I was wondering, should I actually formally applied many places, as you can see here. What do I want to do for the next chapter in my life after it's about 21 years abroad? So there are many SWOT analysis been done. What are the strength and weakness and all these things, that what should I do, so and so. So it come out to be a very long story and very long journey, and it takes about nearly 3 years or even more come out with the joining of this Faculty of Medicine UKM here, and a lot of things around, I say about 4 years or so to plan, and all the form missing, so and so, but I enjoy the journey of that. So the new chapters begin 12th of March, 9 years ago in Faculty of Medicine, and joining the Department of Medicine. And then the first few months, these are in my heart. I was excited. I got a lot of fear of unknown, the new ecosystem, new culture, new friends. And I think people working in silo here. I apologize if these are breaking your heart. There's no dedicated research team. There's no laboratory for me to play around, new in everything. So I get in touch with my mentors. What should I do? Come back and continue that. But he said, wow, you get personal car park first. And consultant there never got personal car park. He asked me to take it easy and study and observe the people. And he said, I'm sure they are observing you too. And try to be kind and have courage. And please, smiling and silencing for the first 6 months of your life in a new place, you see what other people are doing. And let me know how you progress, and keep your networking alive, and go get a big grant to keep you busy and show your endoscopy skills and your capability. So At that time was the first 3 to 4 months or so, still adaptability is very slow, and I remember very difficult to adjust life after so many years abroad. Now, everything, grant proposal, rejection after rejection. I mean, there's so many rejection. I think at this stage I will regard it as a normal physiological process, got rejection from the grant. So I apply, and then these are the remarks from the CRIM. Anda tidak menepati syarat kelayakan permohonan, iaitu pemohon masih belum mengetahui sebarang projek, blah, blah, blah. So what? Who cares? So move on. And get another one, blah, 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 not please. So what? Move on. And also trying to get when my mentor said, try to get the big grants, and I was challenged by someone to write a big grant, and indeed, I put a day and night writing and Googling and so to come out with the grant, but it was rejected, first grant, TRGS. At that time, I don't even have a clue GGPN, what is GGPN? No one tell me all these grants, so I have no idea. So a lot of rejection, the first cycle of TRGS. So analyze what was the issues, and back again, what I can do better. Alhamdulillah, thank God, this is the first grant that I captured for the faculty, the TRGS, asking about 1.2 million, but was given 980,000, it's about 300K deficit. I have to change with all this person, how do we do about it? And I think the first step is always hardest and very difficult, and a lot of, you can see that interference or noise, but you have to move on and on for the properties. So what I'm going to like to spend time with you now is the inflammatory bowel disease, something that close to me for the last many years, attending the clinic abroad, and clinical and research and administrative element of inflammatory bowel disease. So it's a disease, the same thing like a chronic disease like Prof. Nolaila doing for diabetes, or Prof. Nolina in Jabatan Perubatan and neurological disorders. So disease affecting many aspects of life or in the patient. So you can see endoscopically, if you put the tube inside the bowel, you can see this is a normal colonic tissue, but someone who get inflammatory bowel disease, you can easily see a lot of erythema, inflammations, or bad kind of colonic mucosa. So the history of inflammatory bowel disease, I don't want to boring you, it begins for many years. Wilkes and Baruch Crohn that introduced this, and over the time, the disease evolved like any other chronic autoimmune disease, and it's rising steadily also in this part of the world. If you look at the global epidemiology, perhaps in the West, it could be a common etiological agent, but in this part of the world, we can foresee for the last many years, after attending so much discussion with the senior colleagues, so it would be changes, because a lot of changes in the epigenetic regulation, environmental aspect, so in the Asia Pacific region. So the lesson, reflection number 2, that I've learned is that once you go for many meetings, presenting as a participant and speaker, in this time was invited to go to Lausanne in Switzerland, talking about IBD perspective in the aspect of Asia Pacific. So I proposed to colleagues, why don't we write a review? Because in the scientific meeting, all the new data is there in the lectures, all the new data given by all these gurus, excellent gurus in IBD. So we come out with the review articles in the Nature Gastroenterology, Environmental Triggers, Review of Progress and Evidence, and we give ourselves about 7 months to write. And this is quite not that difficult, because we're combining all the slides from the previous speakers, and we see and we divide and we look at all these various aspects of the disease. So if you join any scientific meeting, networking, and you come out with writing some nice review, because at that time people are talking about any latest development of disease at that particular time, and easy to finish out within 4 to 6 months in the motion. And also a lot of reflection, and in particular IBD is susceptible to various influences and continuously to evolve, especially in this part of the world, how the factors, genome, microbiome, and exposome in the pre-IBD, and how can they present late, and what are the factors. So these are the things that can be discussed in many chapters. And also about how the network of the interaction in many aspect of diseases, inflammation in the bowels, in terms of cytokine networks, in terms of not only Crohn's disease, pancreatitis, but also other forms of inflammation. So you can get the ideas a lot for the future to do the research. what you should do with the limited resources availability that you have in the faculty. So we also ask, what's IBD in Asia and Malaysia? If you are invited or give lectures, you give the data from Mayo Clinic, from Stanford, from ever, and you don't have a local data, you don't know what's going on locally. So you can think globally, but you have to act locally. So Jarvis, eventually the students, this is work about 5 years or so, executed very well by Pronafiza and the rest of the team. We come out with analysis of 4 decades of IBD in Malaysia, and we create the registry and prospectively maintain database and to showcase to our peers internationally. And we have shown many other important data in terms of why these cases are raising, even though the catchment area in Churas is mostly Chinese and Malay, but we have observed Indian ethnicity much higher as compared to Chinese and Malay. And also various treatment options that the patient have. And of course, in terms of their disease phenotype, whether they are epidemiological factors, male and females, and education, and these are diseases or the profiling that we maintain until today to renew with various changing of the research assistant, prospectively maintain database. So this is very important. If any of the clinician of your own disease, you come out with your local registry and come out all this, then you will get a lot of things from after that. And it's never been easy because we all know that every patient, not only IBD, but also disease are different. So personalized approach is very important in the future, that when you look back many data that you have in the lab, how we can choose the particular biological agent or targeting various therapies for diseases. Now, Gil Kaplan's very clever guys in Canada have looked at these epidemiological stages of IBD. This is a very recent paper last year, and how he predicted that for the next many years, where the Asia that we are actually involving in this aspect, where so many, we are in the compounding prevalence for the next many years, that excite me, that the data that we have, biospecimen that we have in the lab, and all these clinical details, something that we have to do a lot more for the next 5 years or so. Now, another reflection is your work will sometimes got attention by others. You'll be invited to showcase or to get to write a little editorial about your experience. You see both in the East and the West, and how you're going to predict in terms of the nasty complication of IBD, which is colorectal cancer in the future, and how how we mitigate these diseases that we're going to have in the future. So it's a very, what we call this, you would never know this until you got email to ask what is your experience and showcase your opinions in the disease of your interest. And of course, from the previous word, it's very important that during my course of in Ireland, that during the IBD, you can imagine the boss asked me to look at over 20 years, over 20,000 cases of colon cancer, and analyze how many of them get the IBD. And you can imagine collaborative skills, negotiation skills with the National Cancer Registry, with all other peoples in the public health. You don't even know who are they, but I think you have to have a good communication skills in order to track them, to get a buy-in, and to get the data and show that even the patients with colorectal cancer with or without IBD, they receive the same treatment. So something that new knowledge that we actually produce for this. Now, I think one of the big reflection is international collaboration. Collaboration is very fun, it's always fruitful. So I think it's blessed to have a lot of collaboration in this part of the world from all the Asia-Pacific region, and to tackle disease interests of whatever that your colleagues want to do. And you can imagine this networking and collaboration really powerful, and the consensus, the guidelines, everything that, of course, have a lot of meetings and draft the questionnaire. But I think the outcome is actually very wonderful, and you can be part of the team, so exclusive team in many aspects that you do with all the diseases that you interest, in particular networking with all the great brains, great scientific mind, with the same people who want to drive the research in the university. Now, and you can see the many years that for the next, I can foresee for the next 5-7 years or so, there are many more that we are still doing now, and the output will be come out within the next 3 to 4 years in terms of a lot of aspect that we can do in the area of, in my area of interest in inflammatory bowel diseases. And again and again, not only what we call this networking intrally, in the intra-faculty, inter-faculty, and also nationally, also internationally, various things that you can learn. For example, those who are prescribing a biological agent targeting anti-TNF, it shows also a very good response in the extra-intestinal manifestation for patients who get inflammatory bowel disease, something that we can learn from the group, for example, this Switzerland group cohort. Now, I think as a clinician, many of us like to write the case report. There's no issues. Give a quick win with the case report. But the problem with the case report in the low impact journals, you will not get the citation. So you better think carefully whether you want to write, whether you want to get the quantity of your research number, list of publication. But by the time you want to go to something and you are everywhere, you are case report in the head, in the brain, in the lung, in the kidneys, in the toes, everything. So there is no niche that you have actually. So I'm not a big fan, but actually for the junior researchers, it's quick win, but must be targeted in the big journals for the case report. Now, I think one of the most important lessons is the original article, where you get original ideas after attending many meetings, and you can think what facilities, what technique that you have. And of course, way to go is producing in the high impact journals. It's a different game altogether. It needs a lot of reading. Dedicated yourself to, let's say, I do every Friday afternoon, for example, at least read the abstract of the disease that you love most. You know, in the case of IBD, at least the top 5 journals, what is going on for the last 12 months, or what are the recent weekly journal that you can think about. So I think this is very important. Not only taking your reading and critical appraisal, come up with a proposal, you have a brief executive summary in your head, but most importantly, to get the money. Do not spend other people's money or try to avoid using institution money, but use the grant. Google's nationally, internationally, where you can get the money. And I think it's a very, very important message to me and to all of you in the future undertaking. And new knowledge that will be excite many people. I got many emails from this paper, for example, how we can use high throughput transcriptomic profiling of Malaysian IBD. And this is the first in Asia Pacific. If you can look at the heat map analysis, there are many differentially expressed genes that we can see. And this excited many researchers in the country where they don't have the multi-ethnicity. And, so they will ask a lot of questions, and they ask here and there, and they want to get what they can do for them in the future. So I think it is exciting, something that you choose one disease, again, I repeat, to deep dive and go in the aspect of other etiology, pharmacology, clinical epidemiology, management issues, complication, therapeutics, and so. So I think that is very, very important. And of course, as time goes by, you become a key opinion leaders, and you will ask the opinion of writing something in particular. For example, a lot of clinicians nowadays using biologic or small molecules that we may not know the actual immunological pathways we prescribe, and we don't know the downstream, what are the things that can alter the gene or epigenetic regulation or protein expression. So I think it's very important that this, when we, as time goes by, we learn the experience from our clinical expertise and our basic science, and also how to avoid the use of inappropriate biological agent or small molecules in the future, and many more that we can do. Now, I think if you do yourself, it dies with you, but if you do for others, it will live forever until hereafter. So I think one of the most important aspects as well, sometimes you do one disease, it may be a bit boring. So you ask yourself, what are the areas that you are good at and you have high patience? In other words, you diversify your publication, but keep within your same specialty. You cannot be doing diabetes one day, and you do neurological disorder one day, and you're doing ingrowing toenail next day, and all this aspect. I think try to keep as much as you can in the arena of your specialty to get what is your patients or good at in those areas. So again, I was amazed. I never have any single publication in celiac disease, but few people, colleagues, know that I used to practice in the west of Ireland, where the celiac disease is among the highest in the world, 1 in 300 or so. So they want to get the opinion how the challenges and opportunity once people treated the celiac disease. And it's a pleasure again to write the review with them and to tackle all diseases aspect with all the big gurus in the celiac disease. And do we have a celiac disease nowadays in Malaysia or in Asia Pacific? Something that triggered me after doing some work here, similar like IBD. Now, we also need to go global towards becoming key opinion leaders or whatever expertise you want to do. And of course, nowadays we're talking about many SDGs, climate change, so and so. So we come out with all this, a lot of leaders in the gastroenterology society and how we can use so-called a green endoscopy in the future without wasting, without actually pollute the environment to save the planet in terms of using the appropriate things or the appropriate whatever in the future that you use regional assays. And this also can come out with the publication with the rest of the key leaders in the world. Now, one thing that I learned, I think many of you also try to avoid publishing the same journal. It's not good for your CV because it's telling you that whether you know these editors, whether you know these people, or whether you have no other option to write. So have a diversifications of the high impact journals within your field to showcase rather than targeted the same journal that you actually want to write. Now, I think it's quite important to have a stress every day, because if you have no stress, you become lay back. If you have overstress, you become a burnout. So there are few things that you have some optimal stress to keep you moving from A to B and C, so and so. If not, then you become 9 to 5 person. You become like in the comfort zone, which is not good. You become whatever that you're comfortable with, and you don't want to go beyond your comfort zone to grow more and to grow more in the future. Now, this is a very important lesson that I've learned for the last 7 or 8 years or so, how you bring the industry into research and take a risk and accept a challenge and face fear. Fear means it could be face everything and rise, or it could be fear, face everything and run away from the problem, okay? So I give you one of the examples, and this I've been religiously practicing for the last 7 years or so. If you engage with one of the industry, you have the ideas to do something, and you Google what are the industry that really may be excited to join force with you. In the case is YSP, to do some using a probiotics, probably novel things to treat a Helicobacter infection. You discuss with them, you have executive summaries, your ideas, you meet them a couple of times. This is end of mid-2019. And then do not forget the message, do not forget to put 10,000 or so at least for the publication fees for the company to sponsor you and your student to publish. This is very important because they can budget for you at the very early on so that you don't burden your faculties, your institution to publish in your future publication. So research also teach you how to do the finance, how to do your budgeting, how to do your other skills, you know. So in the case here, let's say 20 something or so plus 10,000, so about 35,000 or so to use for the student. And design carefully the methodology. Takes your time, design carefully. Do not read and duplicate what people do. Do a proper R&D, research and development. Do not read and duplicate. Do research and development. So that, for example, I have to credit to Dr. Izrina because she will be presenting Young Investigator Award in the IDDF, International Digestive Disease Federation in Hong Kong in September, with the hard work and with the right methodology. And this is something that a very big meeting, all the abstract will be accepted and published in the GUT, the leading journal with the impact factor of 33. And importantly, I asked myself, reflection, if in the faculty here, Prof. Tan, for example, if we have, I just put a figure here, 200 clinicians who put 10,000 each per specialist, per industry, we actually save 2 million ringgit of article processing charge for the faculty. This is 100 and how we can save and bang straight away a Q1 article without doing anything. So I'm encouraged myself still, even for the last many years and many more years to come, Industrialization is very important, not only helping to sponsor, but also from A to Z of our work, rather than putting 500 for publication, 300, where you're going to go. So I think this is very important strategies, how we bring the industry to research, but of course, working with industry, a lot of challenge. They're going to get your output, they're going to chase you, you get nightmares, you get this and that, but take it easy, enjoy the ride, and get the strategy right in the beginning. Your strategy, your analysis, your implementation, execution must be in order. You have to be a very organized person. So I think this is a very important lesson that I've learned for the last many, many years. And I think among the industrialization in research and learn negotiation skills and work under pressure, for example, in various partnership with industry that give money to us, to the university, to carry out many research in many diseases, so that we can not just sign the MOU, MOA, what we can bring the money and use for our students, for our faculty, and they also be delighted to be co-authorship in many papers that to come for our faculty. Now, next lesson is, there are many. We should take a planet as a stakeholder. We need to follow the current trending in research. People talking about all the SDGs and climate change, so and so. So you have to go bigger. You have to go more than usual things. So writing the international grant, I have to be credit to Prof. Ahmad Omar, the gastroenterologist in South Wales, in Australia, Prof. Nafiza Mukhtar, Department of Physiology, and Prof. Muhammad Talik Latif, environmental scientist in the FST, how we come out with all this. I still don't understand how the microplastic being processed in the colon. I still don't understand. I saw the picture 3 days ago posted by the researcher, Dr. Madiana in FST, but I still do not understand. So having curiosity, courage, confidence is very important to learn something that grab an international grant for the future, all of you. And of course, there are many other aspects, the politics in research, politics, academia, socio-economic advocacy in health, and how we have to treat patients the same equity health for all these patients that you obsess about to the clinical practice. Now, diagnostic methods in the GI has changed a lot. If you just do the walk round, you just thinking CRP, full blood count, renal profile, liver profile, biochemistry, the same walk round people done many, many 100 years ago. So you have to think a deep dive, how the cells, how the molecular aspect, RNA or DNA, expression protein, whatever aspect can change at the during your round. If you are only just rely on the simple antibody, things like that, it's been going on for many, many years. So if you look at the theme of this year, I would not say new norm, because it could be next norm and next norm, perhaps. You know, what is the new norm next 5 years? It could be new norm again. So it could be what will be the next norm, or the next thing that we need to advance, AI and precision medicine, big data, so and so. So there are many, in fact, things that diagnostic disorders in diseases, I'm sure in many other specialty as well, but of course, multi-omics is very, very important. I'm going to share a little bit citing research that we do in terms of look at not only IBD, but also gut microbiota, probiotics, other diseases that we are doing at the moment. So if someone wants to adventures in gut microbiota or learning new things in research, and also learn new things is also preventing dementia, what evidence said, and we need to know what actually going on. So I think you all are very clever here and reading critically appraised and journals, and what you can translate into your research is very important in the case of, let's say, gut microbiota. Now, the connection between the GI system and other organs, there are many. And for example, in the case of how the gut liver exists in terms of using the fatty liver or non-alcoholic fatty liver disease, or now so-called a metabolic associated fatty liver disease as a model to use for the type 1 or type 2 diabetes and obesity, and what is the goal of the gut microbiota. So we all know prevalence is high. You can read the National Health Mobility Surveys. We're getting more increasing in terms of metabolic dysfunction, obesity, dyslipidemia, and also the data for fatty liver in Malaysia is still lacking at this moment of time, but we know that a fifth of us will have some metabolic disorders. So what we would like to do, how we can manipulate using probiotics as adjuvant therapy for treatment of NAFL in the future, and the beautiful experiments this part of Dr. Najmi doing in terms of how do we learn from the studies that after 6 months of the intervention with the probiotics, the composition of gut microbiota was really maintained. You all learned the alpha and beta diversity in terms of look at the richness species or differences in complexity of the species. You can see in our study here, you can see that the ratio or one of the important dysbiosis marker reveals significant decreasing trend in some microbiota composition, richness density for someone who's been supplied with the probiotics in patient with fatty liver. And indeed, the immune system function, you can see here, there's not only the significant CD8 was reduced in placebo. In other words, those are not having the medicinal product, but you can see here after the analysis, probiotics actually stabilize at the small intestinal system, in which Dr. Najmi did to biopsy and have a look before and after supplementation. You can see here that the probiotic actually increased the expression or so-called these zonular occludence or tight junction after about 6 months of consumption of probiotics. Now, it's actually this idea. I was in Japan attending probiotics. I was smiling myself a couple of days ago. Look at these notes, 2017, tried to modulate this experiment and asked Dr. Najmi to write and how we can do the probiotic in the NAFL and take possibly a biopsy and look at this occluding after attending a scientific meeting. So how attending the scientific meeting in the past, bringing the ideas and what we can do that the limited resources that we have. And indeed, all this shows that there are a massive difference for those who get gut probiotics in terms of maintaining intestinal barrier, in particular zonal protein in patients with fatty liver, try to prove that the gut liver axis is happening, and how can we modulate using the gut microbiota to treat the patient fatty liver rather than traditionally go to other approaches. And indeed, you can see here nicely also that the qPCR analysis, a lot of reduction of the important players, cytokine, in particular interferon gamma and TNF alpha, also no significant in placebo, suggesting that it is working in the future. And of course, the big study need to be done to validate this data. And in fact, what we have seen here is that when you give someone probiotics, you actually give the good gut microbiota, and this produces a lot of good short-chain fatty acid via fermentations, and it will enhance the intestinal permeability of the tight junction in the barrier of the intestine, in this case, duodenal mucosa, and how they eventually improve the immune status and reduce the inflammation of the particular patient consuming probiotic. And these are among the novel finding that we found. And of course, we need to do more to get to examine the true effect of probiotics in the pathogenesis of the fatty liver. And indeed, what we have learned is that combining employing both a clinical master student plus master by research student give a great output. And at the same time, we can raise many other researchers up. So it's not all about you, about your seniors, about you to become correspondent and first author at the time. You raise other people up in your journey. I think that is a very novel and very important and banyak pahala that we get in the future. For example, in the case of this, we get Dr. Najmi, the consultant gastro now, to do this project. And in fact, I have to congratulate to him with the hard work and all this. Now he is ready to swim on his own for the next many years to come. Now, we also look at how the gut-brain axis in the model of irritable bowel syndrome, and we have the student in the past and executed very well by Pranafriza writing the prevalence, how much subthreshold depression among our cohort patient with IBS, a third of them some subthreshold depression. In fact, it is true in clinical practice, and we didn't ask much about this patient. And we want to like the use of this time lactobacillus containing culture and how we can do at the animal model, targeting specific lactobacillus, how they can alleviate or treat mental disorders and perhaps future neurological disorders while they're altering the various neurotransmitter in the brain. So this is designed, credit to Associate Professor Dr. Jayakumar in Department of Physiology here, in terms of grouping the individual's animal model, whether they're in control, whether they're receiving culture med drink, whether they are in the stress and taking probiotics or taking the antidepressant and also stress probiotic plus antidepressant, and put them in the behavior so much so that they are under pressure, for example, restrained or exposed to the light, wet case so, and look at the effect after this. So of course, we have observed exciting things in terms of the changes of the body weight, telling us that this stress treated with probiotics significantly reduced, increased compared to the control. What we also seen is that sucrose preference test, and this one you can ask Dr. Jayakumar how we can, this is a reward-based test, and how the rats undergo a constant unpredictable stress. A treated probiotic have higher sucrose intake compared to only stress or talking placebo. In other words, that there is indicating that the probiotics can alleviate the anhedonia behavior in the animals, and of course possibly also that we're doing straight away the same time with the human model to see whether they are working or not. Now, the force swim test also, Dr. J doing this and looking at if you look at the REX box that indicate the stress rate, and you can see here the stress group a higher immobility behavior. Those who are treated with probiotics as a blue, they get all underwent treated with probiotics, and what the group has reduced immobility behavior, suggesting that also affecting how this manipulation of the gut microbiota affecting the mobility and swimming attitudes among the rats who drinking probiotic as compared to the placebo. So a very, very important findings for us, and of course this is not published yet. And also, if you look at someone who, let's say, rats or even human underwent chronic stress, and normally they go to toilet or panic attack or lose stool and excrete more feces. But when we supply the rats with the higher with the probiotics, they have a lesser fecal excretion as compared to those who are receiving placebo. Something telling that gut microbiota modulation also evolving these properties of cooling down the rat in the study. And also immunohistochemistry, look at the immunostaining of 5-HT and 1A cytoplasm is increased as compared to control. And you can see here that how the modulation with probiotic as compared to control involved in the some receptors. In this thing, we focus on the 5-HT1B and also 5-HT1A in modulating the gut microbiota. So a lot of exciting discussion, and we also do some other works that's going to finish soon, and also some work that we do straight away in the human model. Now, what about the gut microbiota signature in young colorectal cancer? I think we all know that the single commonest cancer for men at this moment of time for the last many years is colorectal cancer. And in women, number 2 after the breast cancer, and it is expected that colon cancer will be number 1 cancer for women for many years to come. And we all know that the cancer is rising steadily. We know that majority of the cases of colon cancer, those who are 50 years or above, but we know that by many years to come, not long while, by 2030, 15% of population will be 60 years or above. And we have observed in practice that there are rising cases of colon cancer among the young, those who are below 50 years old. And we know that on average, as a Malaysian, 1 in 33 Malaysian at risk of the developing colon cancer. And we have done this work for the last 5-6 years prior to publication. And Dr. Najmi has executed very well. I look at over 2 decades or so how this pattern work or not. Is it similar trend the rest of the Asia Pacific countries, or we have behaving something different? We'll look at the incidence and clinical pathological features of colon cancer in the society, in our center. And you can see here the whole lot like MDT team in the team. So what we observe, not surprising, the population, the age between 50 to 75 or so is dominated. But what we see in here is similar trend in many countries in Asia Pacific region. In fact, could be fastest in our region, that the rising of the young colorectal cancer for those who are between 40 to 50 years old. At this stage, we got 11.9%. It used to be about 3 or 4%. Is this represented national data? We don't know, but we have looked at about 1,000 patients of colorectal cancer for the last 20 years or so. Now, so what we observe here is that the cases are rising and the numbers of patients are rising. But what we're interested to know, who are they? Because we know that the colon cancer is highest among the Chinese ethnicity, is known for the last many, many years. But in the study, what we have shown that even 40 to 44 and 45 to 49, the one who get rising more colon cancer is the Malay ethnic group. And this is the catchment area that we see more Chinese and Malay. And now we see the new trend, that new knowledge for us, that the young cancer for the Malay is coming steadily for the last 2 decades and becoming more in the future. So what are the reasons for the current trend? And if you look at in the literature, the incidence of early onset colon cancer expected to rise perhaps more than 140% by 2030. So we all know the adenoma carcinoma sequence of the cancer. They're ranging from the normal colonic epithelium, normal polyp, and they progress to colon cancer. And this takes about 7 to 10 years or so. The many mutations that apply from the beginning of APC mutation until the late gene P53. But there are many other insights in going as well in terms of various, whether this is adenoma carcinoma sequence or this is perhaps could be inflammation drive a carcinogenesis process of this patient that we still don't know. So we know potential risk factor for the early onset colon cancers, straight away, straightforward. Family histories only contribute about 10 to 15% or so. Any other diseases, and also, of course, diabetes, or some regarded as a precancerous, and many other risk factors for the early onset of colon cancer. So there are many, when we do something, there were a lot of questions being raised. In fact, not like we glorify, but we get more issues. And we learn from the reviewers, we learn from the critics, and what we're interested to see in particular, how does the role of microbiome and inflammation have influence on the early onset of colorectal cancer, particularly in Malaysia. Now, so we know that microbiota-associated mechanism in colon cancer, perhaps the beginning of the gut dysbiosis or imbalance between gut and gut bacteria, how they can initiate a cascade of inflammation and turn around to carcinogenesis. Or is it because inflammation first that trigger the gut dysbiosis that leading to carcinogenesis? So this cycle perhaps be repeated and eventually develop colorectal cancer. And we do the, of course, assessment. We look at the patients or early onset. We compare with the late onset, those who get polyp, those who get early onset colon cancer, a normal patient, and of course the matured or early onset using all these assessments or 16S RNA sequencing by Illumina and relate the sequencing results and the clinical findings. So we got a good number of patients, those who get cancers and polyp and control sample. This is a beautiful results that you can see here in terms of alpha diversity. In other words, the richness of the gut microbiota in the different group of the patient. For example, if I go through with you, you can see here a very nice segregation between those who get young colorectal cancer, normal polyp or young polyp. They have a different richness or the composition of the gut microbiota in their polyps or in their cancer in the normal colony. There's something that we found very excited, and I am sure by any doubt this will be published in the high impact journals. And if also we look at the beta diversity, in other words, differences in the complexity of the species of the gut microbiota, you can see a very nice segmentation or delineation between various aspects of the patient, either the young, at the late onset, normal or polyp, where they have a different composition or differences in the gut microbiota, something that we are very, very excited to see. And of course, if you want to go the adventures of the game of the microbiota, you need to know how to isolate the family and genus species using 16S, or of course strain, if you want to get to the shotgun metagenomic to look at different strain of the gut microbiota that you look in the experiment. Now, this is also a beautiful, what we call the relative abundance of the phyla level. If you look at those who, let's say, here is a polyp or even normal people, you can see here the firmicutes, the color here. Firmicutes is relatively preserved in patients who get normal or no cancer or no polyp, as opposed to others who get a polyp or cancer or the late onset cancer. You can see a very nice segregation of the different relative abundance at the phyla level. And similarly, we also see relative abundance at the genus level too. Patients who are normal compared to the polyp colorectal cancer at the young and the late onset. So something that I am sure it will be published again in the very high impact journal. And you can also see at the species level too, where you get a lot of relative abundance of the different species of a different level in terms of a normal patient with the polyp or advanced cancer. And this is a really suitable, really so-called gut microbiota signatures in our populations for the colorectal cancer. And of course, beautiful other things that I'm going to show you in terms of this data. So what we have learned, the microbiome and metabolomic alteration also play a very important in multi-step technique, multi-step development of colon cancer, where a lot of from the adenoma formation, various stages, and how the particular bacteria, for example, Fusobacterium nucleatum, a very highly significant elevation in patients with intramucosal cancer, and various other gut microbiota that we have not discovered and ready to be discovered in the next coming years to come. And of course, a gut dysbiosis or imbalance between good and bad bacteria may induce changes in not only in the genome, but also many immune aspect, metabolomic aspect, or how they can damage or make a destruction at the intestinal barrier function, leading to the cascade, either inflammation first or dysbiosis leading to colorectal cancer. So something that very important, how the interaction or interface between the host and gut microbiota, leading to activations, many cytokine, chemokine growth factors, leading to the influx of all these supposed to be our friends, microorganisms that living in intestinal ecosystem, and how they disrupt and initiate the cascade of inflammation and promote the cancerogenesis in the future. And we also have shown that at the human levels, how we can give the probiotics of patients among those who get colorectal cancer, and we can see very nicely there's a reduction, a lot of important players after 6 months of consumption of probiotic, those who are interleukin-6 or this TNF-alphas or various other cytokine are reduced nicely, and none of them get issues or adverse effect, and how this can probiotic reduces inflammatory cytokines, flares in patients with colorectal cancer, and very, very exciting data. So a lot of conclusion that we have done, and a lot of questions also been asked, and of course this research is still going, and we explore a few more other factors to be done. And this is the work outside the intestinal, and with Prof. Dr. Nolina, how we can work with other colleagues within the same department, and how we can giving the probiotics, and how this can improve the gut motility and constipation among patients with Parkinson's disease. And indeed, again, a lot of questions also been asked, how do we continue this, and how long the duration, what is the actual mechanism, and something that we can work also in the future. And again, with the diabetologists, and how the gut microbiota can be a part of the microbiota signatures in order for us to diagnose someone who get gestational diabetes mellitus, or when they proceed, many of them develop a full-blown diabetes, and how can we improve the gut microbiota to reduce the chances of them to develop a full-blown type 2 diabetes in the future. And of course, a careful consideration for choosing probiotics need a level, in a different level, and you have to discuss with a lot of experts and how if you go to the pharmacy, you will be given 20 different types. What are they, the evidence-based, or are they just for a commercial, because they are not a medicinal product, they are supplement by definition. And I think this landscape will be changed for the next many years. And we also right to do give a guideline to the customers, healthcare customers, in order to choose the right antibiotics. Now, last but not least, the lesson is that medicine, as we know, if you practice 20, 30 years ago, may be different nowadays. We don't do anymore things that we used to do. Medicine evolve rapidly, and so do not forget to update your work. For example, if I write the article last time, many years ago, how we manage gastroesophageal reflux disease, and that time there was no potassium channel exit blockers that can be used to suppress exit inflammation. So I have to update myself. And of course, these are about mentorship and how you get in touch with your mentors and how you can propose to your mentors. You need to update whatever that you have published in the past, so that there are, for example, the use of the PCAP, potassium channel acid blocker, is not in the past. That look at now, things are moving, and there are many more coming for the future. So I think something that we cannot just stay in the comfort zone all the time. Things are evolve. The change is a constant. If you don't change, you will be changed in the future, whatever that you do for your research. Now, a little bit about research leadership. I think leadership is about giving the output. Leadership is about raising others. Leadership is about get the best out of other people. So there are many traits, as you can see in the slide here, from loyalty and empathy and patience and pride and respect each other is very important. But I think research leadership thrives on inspiration, giving inspiration to the young, to the juniors, and not domination. You know, it's not all about you, it's about them and how they're going to travel this journey of research together with you for the future. And I think the great research leader is not best at everything. And he or she will find co-researchers who are best at different things in the molecular biology, in the nutrition, in the engineering, mathematics, and get them on the same research team, and they can do wonders for the research output. And very important, asking myself all the time in terms of performance status, where am I? I think some of you will be familiar with this slide. And in terms of, if you are no publication, no grant, so you need to work on that. But if you are high publication, high grant, you need to help others. You need to mentor others how to get all the things that you need. And this is one of the performance chart that I've learned for the last couple of years for myself and colleagues, how we can do together as a team. Now, if you ask yourself, what is the half-life of the career in clinical medicine? Trust me, I think those who, by the end of your career, most of what we have learned becoming nonsense and not practical anymore. So you can ask your mentors or senior colleagues in the past, they don't use anymore this treatment for the disease A. And there are many phenotypes of researchers or research leaders. Some of them want to happen, they made an effort. Some of them wait to happen, and some of them just wandering around what to happen and waiting for people to trigger him or her to do something. Now, there are many things that you can do to enhance your career in research. You know, not only just determination, dedication, discipline, so-and-so, working and strategy, but in fact, there are many. There are many for you to have. And also I'm saying to myself that you have to good strategies, analysis, choices, and how you execute collaborations, keep journaling and reading and practice. And don't go shopping after attending international scientific meeting. Yes, you can go, why not? You know, this is part of the thing as well. Know the research trending and, you know, and smile and actively listen. We are given one mouth and 2 ears. Perhaps we actively listen more and speak less and do more what need to do. And time management, having curiosity, courage, confidence, compete, you know, a very important, and good communication skills, and monitor the progress, raise the juniors to up different levels, and these are very important. And monitoring research progress is not easy. There are very many kind of progress. You can see on the top bar is also a progress. You can see steadily progress, and you can also see up and down progress. So don't be panic or don't be heartbroken. So everything coming in a nice manner. And we also have to be resilient as a research leaders. Resilient research leaders, very important, how we accept the change, how we become a continuous learner, learn new things, and how we can take charge and finding the sense of purpose and do reflection and lesson what we have learned in the past and what are the skill shift. If you're still talking nowadays soft skills, you are wrong. Because soft skills has been giving this terminology back in 1972, soft skills. Now people are talking about smart and sharp skills. I leave it to you to Google what are they. Soft skills are the out-of-date terminology. So something that we need to learn. And consistent research leadership. You have a consistent ladder to jump from the small grant, medium grant, and this and that. And I think it will be success rather than inconsistent. And of course, work for others is very important, and research leadership and do the best. Always give 100% what you are given the assignment to you and your team. And when you know better, then when you know better and do better, do better, so that people all made mistake in the past. So you need to do better and learn. It's either you succeed or you learn. You won't fail. So get rid of the word failure as part of the learning process that you become a better person in the future. Now, research leader need to do many things. You need to do what we call things to do. You need to choose a different path, and you have a great friendship. And these are one of the snapshots I still doing until today, until this morning. So this is typically I will do this, let's say on Friday, for what next Monday that I need to do. So, so Friday before you log out, before you left, you need to plan your things ahead, what you're going to do Monday to Friday. And trust me, I'm not sure whether you agree or not, we actually may be working 3 days a week. You know why? Because when we're coming on Monday, we just start the gear one, we just bit there, bit here in the afternoon. So we don't actually work on Monday much. So the real working days may be Tuesday, Wednesday, and Thursday. Friday, maybe the weekend, probably coming into our brain already. So we probably commit 50% our work. I do not know who created Saturday and Sunday as a weekend. If I've been created at the time, I choose Friday, Saturday, Sunday as the weekend. Perhaps me, you agree or not? So and it has to be justified to ourselves. Perhaps we have to do a couple of hours work over the weekend because we don't spend much on Monday and Friday. So no harm to do on few hours on Saturday and Sunday or the literature and so and so. And I think, and do not be afraid to choose a different path than others. Sometimes there are many things that crisis that can change to opportunity, as Prof. Harlan said early on. So something that we need to explain or do this religiously. And research leader needs to build a strong research network. You can see that. Focus to the right people, career focus, share values, becoming a connectors between A to B. And remember to reconnect people. Over the corridor, you can say that, hey, I got a researcher very good in this, so and so. Lift others up and create win-win situation. If you're going to profit the time and the effort of other people, it's fair to give return on investment back to them. Don't be selfish. You're going to ask them to meet you, you're going to meet, you want to profit from their expertise, but you got to know that you really have to have a value-based research leadership, create win-win situation, and giving before you receive, and of course, a lot of creating system, sharing things that you should do. And we also have to have a growth mindset. If you always thinking about the problems, complain here and there, not try to find solution, we are not growing. We really have to look the challenges or the crisis is opportunities. We really need to explore something that people has not explored before. If not, we never grow. We'll be like this for the next 50 years. So I think we really have research leaders or any researchers, academician, clinician, scientists, we must have a growth mindset. And remember this, choose one disease that you love and master it well. Build up your own patient database, registry, specimen. Don't rely on other people's words. You send this abstract, you attract industry, you're building your personal branding reputation, you get mentorship, networking, sustainability plan, you join professional societies, aim for the grant, and learn new things. Everything, if I want to learn new things every day, what if you read the book Atomic Changes, by 365 days, you are 37% better than other people. No, just be better. Do not compete to other people. Don't care about other people doing, compete with yourself and do plan what you're going to do. And be humble and build up the interdisciplinary team quickly. And respect and agree to many things. And be careful, ego is the biggest enemy. And never be arrogant in doing not only research, but many things. Try to be a leader, at least for yourself. The success of any group starts from the top, and educate the juniors, promote and lifting others, and many more. And if you do this, you will get the byproducts of research, promotion, ranking, so and so. So you don't need to do that, or I need to do because I don't have one more paper to become associate professor. I need to do this because I need 2 more papers. It will not work that way. These are the byproducts. You do things well, ranking will be raised. You do things good, money can be saved. So I think there are many things, and of course, need to be careful as well. Sometimes people like to joining the research without doing any work, just to get the co-authorship, something that I think we should not tolerate on this. Why people giving up in research? There are many reasons. Of course, it's not rewarding. You will not get, if you are in private practice, you get few thousand in a couple of hours. If you research, you get probably nothing. So these are among the reasons. You want to see the result very fast. You will never want to visualize things are possible. You see a lot of grant rejection, your failures. I'm not good enough. You feel that something that you're going to lose. I think you never ever give up in your career, because that is something that learning a process that we can do. Now, research leaders or academicians or scientists or clinicians need to get out of the comfort zone. We come here 9 to 5, the clinic, we do this, we do that. That is routine after we retire. That's fine. But you will be in the comfort zone all the time. So the growth will be happening. Magic will be happening if you are in the outside your comfort zone. Trust me. So I think it's very important, a message to all of us. And remember this also. This is nicely written by Albert Einstein. Ego is equal to 1 over the knowledge. The more the knowledge, the lesser the ego. The lesser the knowledge, the more the egos. People sometimes like to talk here and there, but in fact, maybe the knowledge is not there. So I think this is an important message that Albert Einstein gave to us. And success in research, it takes a lot of things, sacrifices, failures, criticism, rejection. You have to get a good discipline. Sometimes you have to sacrifice your weekend 2-3 hours to just get the bloody things done nicely without any interruption, for example. So I think it's very, very important. And there are many skills that you need to polish to succeed in research if you want to become a research leader. There are many things: organization skill, communication skill, a research analysis skill, how the previous people who got the grant and what they have done to execute very well, and of course, writing skills, so and so. And the triangle of the success for research leader and research in general, it comes into 3 other your skills. Attitude is very important. If you think you are the smartest in the room, you are wrong. If you go to any meeting, you think you are the smartest people in research, you are wrong, because you need to appreciate other people on the different angles. So you listen and you execute, and of course all the other skills that you have to have as well. And there are many types of intelligence that link to a dynamic research leaders, and are very important. Not IQ, but the EQ is very important because they represent your characters. And social intelligence, very important because represent your charisma. And in this era, these are many, many years ago, adversity question, how we adapt with the pandemic 19, for example. And of course, SQ is also very, very important. A message to me and to the rest: do not waste your time to impress others about your research. It's no point. It's a waste of time. Do what you love in your research of the disease that you love most, and love what you do. There's no point to, I do this, it's a waste of time, and you will not get anything. And something that I learned for the last 4 years or so, secretly, is having a personal board of directors. or becoming a sponsor to the upcoming researcher that you want to train. So they can help you in many things, in coaching, in mentorship, in connector, and also your wellness. And it is very important to have your personal BODs or board of directors in multi-angles in your career in the future. And expect the unexpected thing in research too. There are many factors. The student who do not like to proceed for the research, and then your grant, jealousy, but it's something that in life goes smoothly. You will not call it life because something can happen, but try to change that problems into a solution and learn from the best. Get the best student, get the best brain to join together and do the work nicely. And again, repeating myself, the comfort zone, if we stay here, we don't go to the fear zone, we never learn, we never grow. So get out of your comfort zone and do something that unusual that you have not done in the future. So what do I want to do? Actually, I just want to be a better person for every day and day for the next many years to come. So I have no competition with others. I don't like. I just want to be a better person in many aspects, day to day, for the next 365 days or so. Now, I'm going to read this about few seconds of the video by Paula Nurse, a Nobel laureate in medicine many, many years ago.
If you work too hard, you will keep going in the same direction. And one big advantage of taking some relaxation, to go off and go and walk for a day, not to work all the time in the lab and with your books, is it makes your brain come down and then you come and look at it freshly. People who work just hard all the time never take off the pressure, and it's when you take off the pressure that you can imagine new things. So if you really want to be good, you mustn't work too hard.
Thank you. That's the message. So to summarize many things that I may not have a time to read, the research journey reflections, collaboration is very important, intrally or internationally or nationally, having curiosity, courage, confidence, credential, competition, networking, interdisciplinary team, read, working with industry, targeted grant, be creative and innovative, diversify your research within your specialty. Do not read and duplicate, but do the real research and development. Keep smiling and do your work silently and smartly and let the success make a noise. And of course, we have to pray to God. And I would like to acknowledge many students. I might not be written the names, all of their master students and master of science, PhD students and clinical fellows. They all driving their own world nowadays. And of course, many other people who get involved in this, working together, in particular, spending about a week with the late Prof. Emerita Dato' Khalid Yusof in NUS Singapore with THE, how to learn the ranking and discussion, a really nice experience with him and many years. And of course, a lot of people and give the input, in particular also Prof. Emerita Dr. Wan Zurina and Dr. Asma and previous mentor Larry Egan. And I think these are industry partners. It's a very important. And these are the last slide that I'm going to show you that I think appropriate. I kept this slide. I made it this nearly 22 years ago to remind myself to others that a career progression in life, whatever that you do, that if you are the student, you do internship or housemanship here, a senior house officer, medical officer, you specialize, all this that you have done, do not forget that if you follow the world, if you follow the akhirah, the dunya will follow you. If you follow or you chase for dunya, the akhirah will follow you. And let you live so that you can give benefit to others. You live benefit to others. And akhir kata, yang baik datang dari Allah Subhanahu Wa Taala, dan yang lemah itu datang daripada diri saya sendiri. Terima kasih kepada memilih saya untuk this year, and I think we all working hard. And again, I'm sure there will be success of the MPP, the 24th Medical Research Health Week this week. And of course, I enjoyed, and thank you if this presentation does not get your expectation. Thank you so much, and terima kasih.